Okay, everyone, I need you to open up the packet that you were given yesterday to page 3. This graphic is at the top of page 3. Please pause the video until all students have reached this point. Okay. What you are focusing on in this chart is just the phenotypes and the genotypes for your blood typing. This can donate and can receive is interesting if you care to look at it, but what we need to focus on is right here. So there are 4 blood types that you can have. You could be type O, type AB, type A, or type B. There are actually 6 genotypes that you can have for this though. There's 2 types of dominance for blood typing. You can have complete dominance, which is what we've been working on all along, tall plants versus short plants. And we know that tall is dominant to short. And if you have a tall allele and a short allele, that plant will still be tall due to that complete dominance. You can see that with this lowercase I. So you can have this capital IA allele, and if you have 2 of those, you're homozygous for A-type blood. Or you could be heterozygous for A-type blood and have the capital IA allele and then also have this lowercase I. This is heterozygous, and this IA is completely dominant to the lowercase I. Same thing with your type B. You could have the IB allele, you could be homozygous for that, or you could be heterozygous for type B blood, and you could have the I capital B lowercase I. If you have 2 lowercase i's, that means you have type O blood. Where the codominance comes in is with this type AB. Remember, in codominance, both alleles are dominant, but they don't want to share the spotlight. They both want to be observed. So that's how you get type AB blood, is both of these alleles are expressed, so you get both of these factors in your blood. The next thing I need you to do is we're going to pause the video again. I need you to fill out question number 1, A through G. You need to figure out the genotypes for each person based on the description. So go ahead and pause the video and fill that out now. So let's take a look at these answers. If you're going to be homozygous for the B allele, you need to have 2 B alleles, meaning you would be capital IB, capital IB. Homozygous means the same allele. We have 2 of the same. Heterozygous for the A allele means you would have 1 allele that is capital IA, and then you're going to have lowercase I. Remember, this allele is completely dominant to this allele, so this allele gets hidden, and you'll only have type A blood. Type O blood would be little I, little I. Type A blood, which has a type O parent, would again be heterozygous, and you could Punnett square that out if you were having difficulty with that. Type AB blood, remember this is going to be our codominance, so you'll have an IA, IB, and these last 2 come directly from the chart. Blood can be donated to anybody, meaning that's a universal donor, which is going to be type O blood, so little I, little I. And you can only get blood from a type O donor, meaning you can only receive blood from this person. Again, that's going to be type O blood. If you have questions on this, please make sure to mark it on your worksheet, and I will answer them when I am back tomorrow. So let's move down the sheet, and we're going to work on a couple of these Punnett squares together. So question number 2 says, pretend that Brad Pitt is homozygous for the type B allele and Angelina Jolie is type O. What are all the possible blood types of their baby? Okay, so if Brad Pitt is homozygous for type B, that means he can only have IB, that allele. So his genotype would be this. Angelina Jolie is type O, that means she has to be little I, little I. So now you can see our Punnett square is totally set up. So what is their baby going to be? Well, in every situation, Brad is going to be giving the capital IB and Angelina will be giving the lowercase I. So all 4 genotypes will be the same, meaning the phenotype, the only type of baby they could have, would have type B blood. The next thing you want to do is complete a Punnett square showing all possible blood types for the offspring produced by type O mother and a type AB father. What are the percentages of each offspring? Well, if you have a type O mother, that means her genotype is going to be little I, little I. If you have a type AB father, this is going to be that codominance. So you'll have big I A, big I B. If we cross these two offspring here will be capital IA, little I, and these two offspring here will be capital IB, little I. So percentages, 50% of the offspring will have type A blood, 50% of the offspring will have type B blood. The reason we do so many of these Punnett squares is to get you to notice a pattern. Once you have noticed those patterns and you see that those patterns repeat over and over and over again, it'll help you get through these Punnett squares a lot quicker. And in the case of a genetic counselor, it actually helps them determine the possible offspring much quicker as well. The last Punnett square we're going to look at for this example is question number 4. We have Mrs. Essie, who is type A blood, and Mr. Essie, who is type O blood. They have 3 children named Matthew, Mark, and Luke. Mark is type O, Matthew is type A, and Luke is type AB. Please pause the video now and try and finish question 4 on your own. Once all students have completed question 4, you can start the video again and I'll give you the answers. All right, so if Mrs. Essie is type A and Mr. Essie is type O, their genotypes should be here. Mrs. Essie is type A, but we don't know if she's homozygous for type A blood or heterozygous for type A blood. So she's going to get the capital IA allele, and then we're just going to leave her second one blank for right now. Mr. Essie is type O, so he can only be little I, little I. They have 3 children. If we look here and we mix this column here, we cross this column, both of these children will be capital IA and then the lowercase I. That is going to cover Matthew over here for his type A blood. This one over here, we have another son that could be type O and another son that could be type AB. Well, if Mrs. Essie has type A blood, the only other piece that can go over here is this type O, which means we don't not know what she is. She's got to have I big A, little I. So this would be Matthew's blood, and this would be Mark's blood. But Luke can't be their child because nobody has a B allele to give him. Luke would need to have the I capital B allele somewhere in his blood, and he didn't get that from either of these parents. So Luke is not their child. All right, let's move on. Go ahead and turn in your packet to page number 7. We're going to talk about sex linkages really quickly. I know we did this in class already, but I wanted to review, okay? This is looking at hemophilia. And I want you to remember that there's a difference in sex linkage because the X chromosome is bigger than the Y chromosome. So down here at the bottom of the X chromosome, these are where the hemophilia genes are. Females, because they get 2 of these X chromosomes, will get 2 alleles for hemophilia, which is why they'll either be X big H, which would be a normal female. She has normal blood. She could be a carrier, X big H, X little h, which means she has the hemophilia allele, but this is completely dominant to this allele, so she has normal clotting blood. And this would be a female who's a hemophiliac. She would have 2 copies of the recessive gene. Remember that hemophilia is a blood clotting disorder. And generally these humans don't tend to live very long because they have a really rough childhood. Think about all the times you fell down and got a bruise or cut your knee. People who have hemophilia, they can't form blood clots. So small injuries are really, really dangerous to them. We do have better medicine nowadays, but this gene, generally, if you were a hemophiliac before 1950 or something, it was very fatal. Boys, you only get 1 copy of the gene. You get an X chromosome from your mom and you get a Y chromosome from your dad. You only get 1 allele for hemophilia because you can see over here on the Y chromosome, you don't have that copy of the gene. So you have a 50-50 chance to get this trait. So remember where we said this female here, she got 2 copies of the gene. She has hemophilia. Over here, guys, you can either get the dominant gene and a Y, or you can get the recessive gene and the Y and have hemophilia. So that's how this works. Please remember when you're doing these crosses that you need to put the X and Y. So if we have a man who has hemophilia, you need to put both of these chromosomes, X little h, Y, and a woman who is a carrier, X big H, X little h. You need to put both pieces down. All right, the last thing I'm going to talk about today is dihybrid crosses. This is in your big packet in your that I gave you that has your PowerPoint notes and your video notes in it. The notes for this are actually on page 8 of the packet I gave you yesterday, so you can follow along in either place. The first piece that you need to do is write the genotypes of both parents. So they'll say they're homozygous for this or heterozygous for this, and you have to kind of figure out what their genotypes are. So in this case, this parent is purebred for both the dominant alleles for A and B, and this parent is purebred for the recessive versions of each alleles. Now you're going to use the FOIL method to figure out what goes where. Instead of a simple 4-square Punnett square, this ends up being a 16-square Punnett square. So how does this go? Well, FOIL stands for first, outside, inside, last. So remember, in meiosis, these don't have to line up in any particular way. A is going to line up with A, and B is going to line up with B. And then what you get next is a mix. So you could get the first A and the first B, and in this case, it would just be capital A, capital B. The next one, you could get the outside A and the outside B, so that again would be capital A, capital B. You could get the inside A and the inside B, so that would be capital A, capital B. Or you could get the last A and the last B, and that would be capital A, capital B. You do the same thing for this parent over here. So the first A and the first B are both lowercase. The outside A, the outside B are both lowercase. The inside A and the inside B are both lowercase. And the last A and the last B are both lowercase. Okay, now let's move these from here and put them over here on our Punnett square. My screen kind of cut off the last 8 of these, but you're going to see that this Punnett square is pretty simple. So now we cross these. So this offspring here would be big A, big B, little b. There we go. We're going to do the same thing over and over and over and over. Big A, big B, little b. Click. Big A, big B, little b. Click. Oh, they're all the same thing. Okay, so that's how you're going to run your Punnett squares for the dihybrid cross. This video is on my website, so if you need to go back and get some more instruction on how to do this, this video is there for you to watch over and over and over again. Today in class, you are working on finishing all the worksheets in your packet with the PowerPoint notes and the videos. So that packet you're working on finishing and completing, and you're also working on completing the packet I gave you yesterday. If you have questions, please email me. These will be due at the end of class tomorrow, so the end of class on Thursday. Again, if you have questions, email me or go watch the videos again. Good luck, and I'll see you tomorrow.